Okay. Thank you, Katie. Let's begin. I'm pleased to introduce you Dr. Katherine Ryan Amato. Katie, as we used to call her, holds a Bachelor of Arts degree in biology from Dartmouth College. She received her PhD in ecology, evolution, and conservation biology from the University of Illinois at Urbana-Champaign in 2013. She completed a postdoc at the University of Colorado Boulder, focusing on the gut microbiota of non-human primates, with a particular interest in determining how the gut microbiota may have facilitated the evolutionary transition of leaf-eating primates to the consumption of hard-to-digest resources. She is currently an associate professor in the Department of Anthropology at Northwestern University in Chicago, where she studies how the gut microbiome contributes to human and non-human primate health, ecology, and evolution. Her research focuses on understanding microbial interactions with host diet, energetics, and metabolism, using life history theory as a guiding frame. Katie is a world authority on these topics. So Katie, please tell us about your amazing work on the gut microbiome.
Thank you. Thank you so much for having me, Dr. Bica Marquez, Julio. It's a pleasure to be here. I wish I was actually there. It's kind of a rainy, chilly day in Chicago. But I'm happy to talk a little bit about what I do. To start, I try to think about how we can integrate the gut microbiota into our understanding of host evolution and ecology. And I think this is an important complement to a lot of the more biomedical research that happens in the gut microbiome and has implications not only for understanding host ecology and evolution, but also for furthering our understanding in medicine and health. So I think about the gut microbiome from the perspective of life history. So as many of us know, life history involves understanding how an organism will get its energy and nutrients from the environment. And then we'll allocate it to things like maintenance, growth, or reproduction. And depending on how hosts determine the way in which they're going to divide energy and nutrients between these different processes will influence host survival and reproductive success, and ultimately will contribute to patterns in host ecology and evolution. Now, traditionally, we have thought about host life history from the perspectives of understanding shifts in host physiology and behavior. So for example, if the environment changes, maybe there's less food available, so that there's less energy, right? Then hosts will have to decide how they're going to allocate energy differently. Maybe they'll become less active so that they can maintain processes like growth and reproduction. Or if something like the amount of energy that's necessary for growth and reproduction increases, say a female is pregnant, perhaps an individual will have to be less active or consume more food in order to maintain these balances. Now, so we know that these are behavioral and physiological shifts, but research over the past few decades has shown that the microbiome can interact with this, both in terms of determining how much energy an organism can get from the environment. So we know that microbes help break down compounds in food like fiber that hosts cannot digest themselves, and they can produce energy. So it's an actual energy source that hosts would not have access to without microbes. We also know that these microbes interact in determining how hosts allocate energy. They can actually determine whether hosts use energy, store energy, what sorts of tissues it's going to. And so I'm really interested in kind of understanding how microbes interact with this whole process and can kind of answer questions about it. Now, just to make this graph a little bit more confusing, I'm particularly interested in kind of comparing what's happening, either between species or within a species across habitats, or even within an individual over time. And so you can see, right, if the amount of energy that's changing here, that's going into the system is more on this side compared to that side, then you're going to have differences in how energy is allocated, and then you'll have differences in survival and reproduction, host ecology, and evolution, right? And so my research group is interested in kind of all parts of this figure, right? Understanding how microbes are interacting with all of these processes. But today I'm going to primarily focus on this first part, trying to understand how the gut microbiome is interacting with host's ability to extract energy from the environment and or kind of buffer environmental changes. And so the overarching question I really want to address here is, does the gut microbiota help buffer wild primates in this case, but animals more broadly, against nutritional challenges? Now, when I started to do this research, I was very much focused on non-human primates, and I wanted to understand a system that would be good to do this. And I chose howler monkeys, the genus Alawata, because howler monkeys have a very flexible diet, right? As I'm sure many of you know, during periods when there's fruit available, the howler monkeys will eat the fruit and their diet will be made up primarily of fruit. And when there's not fruit available, they'll shift their diet and consume more leaves. And we know that these food types are very different, right? Fruits tend to have higher concentrations of sugar, energy that's easily accessible by the host, and they tend to have lower amounts of things like fiber and secondary plant metabolites, whereas leaves tend to have more fiber and plant secondary metabolites and less easily available energy. However, we know that howler monkeys, generally speaking, maintain kind of their behavioral profile across the year, depending on the site. And it's not necessarily that when they eat more leaves, they are actually less active. And so there's something that's helping them to kind of meet their energetic demands despite the shift in dietary digestibility, essentially. And so this is a really interesting system for looking at this. Also, as I'm sure many of you know, howler monkeys are widely distributed throughout Mexico, Central America, and South America. And so they're also a really interesting genus to look at because we can really look at these dynamics across different habitats and in different species. So when I decided to start doing this, I started by focusing on the black howler monkey, Alouatta pigra, in southeastern Mexico in the state of Chiapas at Palenque National Park. And I was interested in understanding this question about seasonality and diet and whether the gut microbiota was helping howler monkeys deal with the nutritional challenges that they were facing seasonally as their diet changed. So to do this project, I collected 10 months of data from two groups of howler monkeys. So this was behavioral data describing diet and activity, as well as fecal samples that I used to generate information about microbial taxonomic composition in the fecal samples. This is a proxy for what's in the gut. We did this using 16S rRNA gene amplicon sequencing. And then we also use those fecal samples to test for concentrations of different metabolites produced by the gut microbiome. One of these is short chain fatty acids, or SCFAs. These are compounds that are produced by microbes when they degrade fiber and to some extent protein. And they're used by hosts as an energy source, right? So they can be directly extracted from fiber by microbes, and then they're used by the host as energy. So we looked at these concentrations to try to understand what kind of amounts of energy the microbes were producing for the howler monkeys, as well as looking at kind of the other kind of metabolites that these microbes were producing as they worked. So what we found was that microbiome composition in these howler monkeys differs across seasons. Each point on this plot represents the average microbiome composition of one individual howler monkey during a season. There are 3 seasons, right? Black is wet fruit dominated. So it was a rainy season where the diet was more heavily based in fruit. The dry leaf dominated season is in gray, and the dry fruit dominated season is in white. And the closer together points are, the more similar the microbiomes are, the more farther apart, the more distinct. And so very clearly here you see that the microbiome of these howler monkeys is distinct across seasons in terms of its overall composition. Now, is this related to diet? It is. So when we actually compared the two matrices for microbiome composition and diet composition over time, we found that there was an overall correlation between the microbiome and diet composition. And there were many pairwise associations between different aspects of diet and different microbial genera. And this little graphic here, things that are linked in blue have a positive association, and things that are linked in red have a negative association. So you can see that we found relationships both among kind of the plant parts the howler monkeys were consuming, as well as estimated amounts of things like protein, lipids, and fiber that they were consuming. The last thing I want to point out is that we saw differences in the two major families that tend to degrade carbohydrates in mammals, the two microbial families, Lactospiraceae and Ruminococcaceae. And these actually shifted across seasons, but they were more associated with energy intake. So interestingly, beyond kind of just fruit versus leaves, as the howler monkeys as I sampled them across seasons, the amount of energy that they could metabolize themselves without the microbes decreased as I moved across the year. And you can see very clearly that these two major taxa that are associated with carbohydrate degradation are also shifting as that energy intake changes. So the microbiome is changing in composition. It looks to be associated with diet. Is this doing anything with regards to function? The answer is yes. So here on the left is an ordination plot. These are the metabolites that were produced, kind of just all of the metabolites. This was an untargeted metabolomics analysis. And again, the idea here is just different seasons are different. You can see that they're clustering very differently. So these microbes are doing different things across the different seasons. And when we look here on the right, these are the short chain fatty acids. You can see very clearly that as we move across this period, across the seasons from wet fruit dominated to dry leaf dominated to dry fruit dominated, we see a change in short chain fatty acid production, particularly during this dry fruit dominated period. And so during the period when energy intake is lowest by the howler monkeys, the microbes are producing the most energy. So altogether, these data suggested that the gut microbiome is shifting in its composition and its function across seasons. These shifts are correlated with changes in diet, and the shifts are things that we think would be benefiting host nutrition. So this was a really good kind of first evidence to try to see if these microbes are actually helping their host mediate these environmental changes. Of course, there are outstanding questions. Is this generalizable? Is this something that happens in other primate species and other animals? And is host energy balance actually stable, right? We weren't actually measuring host nutritional status or energy balance. So we're just seeing patterns in what's happening in terms of diet and the microbes that suggest that host energy balance is likely stable. One thing I should mention is the howler monkey activity budgets were mainly unchanged across the seasons, aside from a lot of resting during kind of the hottest period of the year, which seemed to be associated with thermal regulation. So to kind of try to take a next step in answering this question, I formed a collaboration with Martin Kowalewski in Argentina to try to look at a different species of howler monkey, the black and gold howler monkey. And so we're now at the opposite end of the howler monkey distribution. And what's interesting about the black and gold howler monkeys is that they live in an even more seasonal environment than the black howler monkey in Mexico. So these howler monkeys are located here. They're studied at the Estación Biológica Corrientes in northeastern Argentina. And there are two populations that we were looking at, one that's around the field station on the mainland, and then one on Isla Brasilera, which is a riparian island that is a little bit more buffered in terms of climate. So we're pretty far south here, so we're seeing a lot bigger changes in temperature across seasons as well as food availability. And this has been shown in previous studies.
We are not listening to you, Katie. Maybe you are mute for some reason. Let me see what may have happened. No, you are supposed to be not mute, but you are, or I don't know why.
Yeah, check your microphone, see if anything happens. It just stopped working.
Huh.
Okay.
Now it's back.
Oh, now it's back?
Yeah.
Yeah, okay.
Now it's back.
Weird. You know, okay. I have some of the like headphones and sometimes they kick in even if I don't have
okay.
them on
It
here.
was just like a minute or two. Now, 1 minute probably.
Okay. So we're in Argentina looking at black and gold howler monkeys. And so really what I was just trying to say here is that looking at diet changes across seasons. So here this top is fleshy fruit availability, then mature leaves and new leaves here. The two different sites right on the island in the dark band and the mainland in the dashed here. And really the pattern is that again, we're seeing high seasonality, particularly in fruit consumption at this site. And so we wanted to see if we see the same sorts of microbial patterns here in the black and gold howler monkey that we see in the black howler monkey. Oops. So our approach here was also to collect data over 13 months. Like I said, this is a collaboration with Martin Kowalewski, but also Aaron Vogel and Sahana Kutyar. We collected fecal samples to look at microbial taxonomy across seasons. We also again did the fecal short chain fatty acid concentrations. And then with Aaron's help, we also ran assays for urinary C-peptide. Urinary C-peptide is a proxy for host energy balance overall. It happens when insulin gets broken down in the body, it's produced, and we can measure it in urine and kind of get a feel for how much energy balance may be changing in the host. So this is a little hard to look at because it's broken into 4 seasons, but this is what kind of the overall plot looks like. And I should put circles on here, but you can kind of see over here, the spring and summer points are tending to cluster over here, and the fall and winter are clustering over here. So we did statistically see a difference in microbiome composition across these different seasons in the black and gold howler monkeys. When we looked at the short chain fatty acid concentrations in the fecal material, so that's that energy compound produced by the microbes, we saw differences, particularly in the spring season. There were much higher concentrations of these short chain fatty acids. And what is interesting and nice to see is that this overlays with previously collected data about the diet, right? This is exactly when there's less fruit being consumed and more leaves being consumed by the howler monkeys. So we're seeing a kind of similar type of pattern that we saw in the black howler monkeys in Mexico. We're seeing more short chain fatty acids produced by these howler monkeys in Argentina during the periods where they're undergoing the most thermoregulatory stress and dietary stress. And when we looked at the C-peptide values, we also saw that there was no change. So essentially the energy balance is stable over time despite these changes in temperature, food availability, and then the microbial composition and function are shifting kind of in response to this, it looks like, and we're seeing that energy balance remaining stable over time. So in black and gold howler monkeys, this suggests that, again, gut microbiome composition and function is shifting with seasons, and these changes again appear to benefit host nutrition. So we're seeing a very similar pattern here. And so overall in howler monkeys, it seems like the gut microbiota can buffer against temporary nutritional challenges, so these seasonal shifts in food availability. An important consideration, however, when we're thinking about the microbiome, is that each species of particularly mammals has its own microbial fingerprint. And so these are data, these are again from primates, but we see similar sorts of patterns in other mammals. We sampled 18 species of primates in the wild from around the world to try to understand if diet or host phylogeny was more important in determining the composition of the microbiome. These points are somewhat small, but triangles versus circles represent a different dietary strategy, so more leaf eating versus less leaf eating. And then the colors are kind of broadly described groups of primates. Catarrhines in the old world monkey category, apes, platyrrhines in the new world monkey category, and lemurs. And you can see again clearly on this ordination plot, these are really clustering by who you are phylogenetically as a primate species rather than what you are consuming. And so diet matters when we're looking at the single species. We see seasonal differences, but the magnitude of those changes is much smaller than the magnitude of difference between any given host species, right? So phylogeny is a bigger effect than diet. And so really what we can say is that these different host species have microbial fingerprints that are very different from each other. And so the question then becomes, if we have these very different microbial fingerprints across host species, do those microbial communities that are specific to different hosts interact the same way with the environment? And so we saw with howler monkeys within that genus, it seems like things are operating very similar, at least in these two species. And you can see them all kind of clustering kind of over here in the platyrrhines. But what happens if we look at something totally different, like a lemur down here? So I just want to kind of go briefly through some of the data that we have in terms of looking at whether seasonal patterns in the gut microbiota are similar in other host species. So I'll give you a few examples from studies I've been involved with collaborators. The first is from Shafak in Madagascar. And here you can see these are relative abundances of different phyla of microbes across different months. And we did find significant differences in the relative abundances of different microbial phyla over seasons and also overall microbial composition. So in these Shafak, which are leaf-eating specialists that have a very seasonal environment that they live in, we are also seeing these sorts of shifts. If we look at geladas, which live in the highlands of Ethiopia and focus on grass, we see a similar story. So this is dry season versus wet season, and again, the relative abundance of different bacteria phyla. And if we look at overall composition, the story is the same. We see differences in the microbiome in the dry season versus the wet season, and we think this is again linked to differences in the types of grass they're eating, the different parts of the grass that they're eating when there's more or less rain. So this seems to be holding up. However, data that we have from white-faced capuchins suggests this is not always the case. So these are white-faced capuchins in La Suerte in Costa Rica, which is a relatively aseasonal forest, at least in terms of food availability for these capuchins. And you can see there's kind of really slight changes in the different phyla of microbes across seasons, but these are not statistically significant, and they are really very slight if you even just look at this graph. And so we're really not seeing these shifts in these capuchins over time. And so the question is this because the forest is just not so seasonal and then their diet doesn't change all that much, or is this something specific about the capuchin gut microbiome? So I'll show you data from a completely different research group that suggests it has to do with the habitat, right? So this is data from white-faced capuchins in northwestern Costa Rica, in Santa Rosa, which is a tropical dry forest. So they have a very seasonal diet in this habitat as they go into the dry season and all the trees lose their leaves. And you can see again, the different kind of colors and shapes here are different seasons. And if you look at this ordination plot, there's very big differences in the gut microbiome across seasons, which suggests that these capuchins are having these big microbial shifts that go along with their dietary shifts. So it seems that there may be these unique responses of the microbiome to environment, but clearly it also depends what environment you're in, right? If your diet's not changing that much across seasons, your microbiome is probably not changing that much as well. One other kind of part of the story that I'd like to add has to do with kind of dietary niche in general. These are data that were generated by the Amboseli Baboon Project. So these are baboons in Kenya. And so what you can see, this is a little hard to look at, but this on the bottom here is days between samples. So they basically measured how different the microbiome was if you collected samples 3 days apart, 5 days apart, 100 days apart, and they have many, many samples from many years, so they can go up to a very big span of days between samples. And this on the y-axis is basically how different they are from each other. And what you should be seeing here is basically no pattern. It doesn't matter how far apart you sample baboons from each other. The samples that are collected 2 days apart can be just as different as samples that are collected 2 years apart or 2 seasons apart. And so we also don't see this seasonality in the baboons. But I suspect what's happening here is that baboons are highly omnivorous primates, right? So they don't have as much of a seasonal change in their diet. They have more to day-to-day changes in terms of what they happen to find on a given day and consume. And so you're, again, not seeing the seasonal pattern. You're more seeing that the microbiome has to be flexible potentially from day to day as these baboons consume different things. So kind of this broader picture seems that, you know, there is evidence that the gut microbiota can help buffer, let's say, howler monkeys against temporary nutritional challenges. And this relationship looks like it's occurring across other species and habitats, but it does seem to depend on the dietary niche and the ecosystem. And so we really need to collect better data for primate species in general and other mammal species to try to understand when are these relationships occurring and in what context, and when are they not occurring, and what are the sorts of dynamics that are determining those differences. The second part I want to talk about here that involves the non-human primates is, can the gut microbiota buffer against chronic nutritional challenges, right? So if the microbiome can help a howler monkey or any sort of primate get kind of extra energy out of its food during a season when there's less food, what about when we're thinking about, let's say, habitat degradation and the dietary changes that introduces? Can the microbiota actually help there as well? So for this, I have some data that are, again, from the black howler monkeys in Mexico, but they're collected not only from the more continuous forest in Palenque National Park, but also from places like a nearby hotel that has a forest fragment associated with it, a nearby sanctuary, and some other forest fragments in the area. What happens then? Do we see the same sorts of shifts? These are older data, so the graphs aren't quite as nice, but this is a dendrogram here. It's kind of like the ordination plot, just showing similarities and differences between overall microbiome composition. Over here is the kind of more continuous forest. This is, the continuous forest is the black here. This is a semi-deciduous but relatively continuous forest. The forest fragment over here is in brown, and then the captive environment is in gray. And again, you can see the microbiome composition is very different across these habitats. This is an assessment of microbial diversity. And so it's basically kind of how much diversity do you accumulate as we include more DNA sequences from these microbes. But you can basically see that up here we have kind of the more continuous evergreen rainforest. Howler monkeys have higher microbial diversity, and it starts to decrease as we get into different habitats. And this seems to be associated with dietary diversity. In fact, when we actually look at dietary diversity in each of these habitats, there is a correlation between the diversity of food items that the primates are consuming in that habitat and the diversity of their microbiome. So the microbiome doesn't look like it's kind of buffering in this case. More so what looks like is happening is that as dietary diversity decreases, so does microbial diversity. And we think of decreased microbial diversity as being a potential health risk, just like we like increased biodiversity in our macro ecology systems, we also like to see that in our micro systems. And so we don't think this is a good thing. On top of that, the shifts that we're seeing look like they're shifts that are not good in terms of the microbial taxa. So as we move kind of from the more continuous evergreen rainforest to semi-deciduous to fragmented forest to captivity, this kind of reduction in dietary diversity, we see a reduction in microbial taxa like Butyr vibrio, which is one of those short chain fatty acid producers that can produce extra energy for the host. And we tend to see an increase in microbial taxa like D-sulfovibrio, which as part of their metabolism produce toxic hydrogen sulfide gas that has been associated with things like colitis and kind of intestinal inflammation more generally. So these shifts that are happening in response to kind of chronic diet change across different habitats look like they're actually negatively impacting the gut microbiome and kind of outpacing its ability to buffer against these sorts of changes. So again, the question is, do these responses hold up when we look across species? This is a study, pardon me, where we compared the microbiome of the black-howlered monkey and the mantled howler monkey and their responses to habitat fragmentation. So we have evergreen rainforest and semi-deciduous forest in this study. And then both of these howler monkey species. These are considered sister species. They're very closely related, very similar ecologies, but they do have somewhat different microbiomes. So the question is, do the microbiomes respond differently to forest fragmentation when we compare it kind of across these different forest types? We already saw that the black howler monkey has this kind of significant negative impact on the gut microbiome. Do we see this in the mantled howler monkey? Overall, the answer is no, we don't really see this as strongly in the mantled howler monkey. So I put these ellipses on here so you can kind of get a feel, but for the black howler monkey, as you move from kind of the more primary forest to the secondary forest, you do see these shifts in the microbiome. And you can see very clearly there's this kind of farther clustering here. That happens in both the rainforest and the semi-deciduous forest. But in the mantled howler monkey, while there are shifts, they're much smaller, and in some cases they're not significant. And the effect size of these, they're close, but they are different, and we're seeing a smaller effect size in the mantled howler monkey. So this suggests that, you know, different species may respond to the same environmental challenge differently in terms of their microbiome. And why I think it's interesting to think about this is, while I told you that these two howler monkey species are very closely related, they actually have very different geographic ranges. And interestingly to me, the mantled howler monkey is the one that had kind of the least negative impact of habitat change on its gut microbiome, suggesting its gut microbiome may be able to continue to buffer across a wider range of habitats. And this is the species that actually has the wider geographic distribution. And so I'm really interested in exploring further these ideas of whether this relationship between the host microbiome and the environment and its ability to kind of buffer against dietary challenges could be involved in part in determining species distributions. So there are clearly constraints on the ability of the gut microbiota to buffer against these chronic dietary changes, right? It seems like in these temporary kind of seasonal situations, you can have the gut microbiota stepping in and helping to buffer. But once those changes become more chronic and you're seeing reduced dietary diversity, that actually appears to have a negative impact on the microbiota. And it seems to actually impede its ability to kind of do this buffering, even seasonally. And again, this is likely the case across species and habitats, but this is currently a pretty open question. And it likely depends on the host microbiome, you know, what that microbial fingerprint is, the dietary niche, and the ecosystem that you're finding these species in. And again, I don't think this is unique to primates. I think this is something that's probably occurring across mammal species or even animals more broadly, but we just don't have good data right now to really be able to kind of bring this all together and get a comparative perspective. So I think that's something where we can really build on this research moving forward. Okay, so for kind of the last section of this talk, I want to focus on humans a little bit. So what about humans, right? I've shown that different primates have different microbial communities that can interact with their environment differently and are probably influencing ecology and evolution via impacts on things like life history. Do we think this is occurring in humans? Well, kind of one of the first questions that we have to think about is kind of uniqueness in humans compared to other primates. All primates have their own unique traits, and if we're thinking about humans, or all animals have their own unique traits, If we're thinking about humans in the context of life history and kind of ecology, there are some really interesting unique traits. We're the most widespread primate on the planet, which means we're exposed to all sorts of environmental variation across time and space. And this is true now and certainly was true, potentially more true for our ancestors. And we also have expensive life history traits, right? So we have things like relatively large brains, short birth intervals, and a long lifespan, which means we have to invest a lot of energy over long periods of time when these kind of environmental changes are occurring. We are certain to encounter environmental changes that are going to impact our ability to get energy. So as a starting question, I'm interested in understanding if the human microbiome is unique and if there are unique aspects of the human microbiome that can be linked to the unique aspects of our ecology and evolutionary history. So to answer this question, I pulled data from those wild primate species I showed you before, the 18 species, and compared them to data from many different human populations that had been published. And what was interesting here is that it suggests that in some ways humans do have a unique gut microbiota. So if you look here on the left, phylosymbiosis is what we often see between particularly mammals and their gut microbiota. And this means that if we make a tree with the host genes and we make a tree with the microbial genes from those hosts, the trees will match generally. We see these patterns of phylosymbiosis. We see this in primates, absolutely. However, humans seem to be an exception. So if we look here, these are data from apes, these are chimpanzees and gorillas. These are data from humans. They're humans that are mostly engaging in kind of hunter-gathering or horticulture, agriculture. They eat fewer processed foods. The pattern is the same if we include humans from other populations, but it's just a little clearer to see with humans that are not consuming a lot of processed foods. And then down here are cercopithecines. So these are things like baboons, red-tailed monkeys, etc. And you can actually see that humans very clearly are breaking this phylosymbiotic pattern. They're clustering closer to these cercopithecines than they are to the genetically more related apes. This is true both in terms of microbial composition and overall potential function. Okay, so humans are breaking this pattern, and we think this may be associated with our ecological histories. So chimpanzees and gorillas are more genetically related, but they have diverged to be more specialized for living in forest environments. They tend to have a more plant-based diet. Chimpanzees in particularly are highly frugivorous. They eat a lot of fruit. And that's not the human evolutionary history. We evolved, we believe, in these open woodland environments with a highly omnivorous diet that included plant parts, meat, etc. And our physiology shows that. We have physiological differences in our digestive tract that are different from those of other apes because of this evolutionary history. And the non-human primates that tend to inhabit this kind of niche currently, and again, evolutionarily, are things like cercopithecines. And so it seems that the human microbiome has converged to be more similar to that of these cercopithecines, and it has diverged from that of other apes. So we're breaking this this phylosymbiotic pattern that we see in other primates. What's also interesting is that in humans, this is a plot. Again, each point is an individual human or an individual non-human primate. We have much higher inter-individual variation. This is in microbial function compared to that of other primates. So this is basically how much difference you have between any given human individual and kind of the average in the center here. And this is the same for other primate species. And it looks somewhat similar. There is slightly bigger distances for the humans. But what I want to draw your attention to here as well is this actually includes chimpanzees, gorillas, baboons, and red tails in this kind of non-human primate thing. And so we're seeing just as much variation between individual humans as we are seeing between individual non-human primate species that cross species. So there's a lot more variation in the microbiome across humans, which could have made us kind of more plastic and able to deal with seasonal environments, evolutionarily speaking, and clearly in modern populations. So I think this is interesting to think about in terms of our ability to kind of buffer against nutritional challenges and be able to adapt to local environments. I also think that it's interesting to think about short chain fatty acids in this context. Short chain fatty acids are a source of additional energy that you can get out of your food. And since we know that humans have a high energy requirement, it's really interesting to think about whether microbes could have helped contribute to that by actually providing more short chain fatty acids, right? We tend to think of things like hunting, cooking, perhaps I think fermented food use is important here in terms of making the human diet more digestible and high quality, and that could have contributed to our ability to energetically support large brains. But I think if we consider microbes, it may also add another important piece to the puzzle, because things like cooking don't actually line up exactly well with the timeline for encephalization in humans. However, so it's likely that humans, I would say, depend more on short chain fatty acids compared to other non-human primates. However, because of all the food changes that I just mentioned, things like hunting, domestication of foods, cooking, we have had a lower fiber diet than other primates for a very long time. And if we think about short chain fatty acids, at the very beginning, I said we get those from microbes breaking down fiber. And so if we have a low fiber diet, is it possible that humans also are not as good at producing short chain fatty acids because we've adapted to a different diet that makes it more difficult to produce those? So to start to look at this, we wanted to look at short chain fatty acids in humans compared to primates. There are not good comparative data on this currently in actual fecal samples. So what we decided to do, I did this in collaboration with Liz Malat, who was my postdoc at the time. We actually went in to look for the genes that are associated with the production of short chain fatty acids. And particularly we looked at butyrate because it was kind of a gene we knew enough about, but there's enough variation in how many microbes produce it. So we looked at 4 different pathways for butyrate production here. Interestingly, only one of these pathways actually starts from fiber. The other 3 are pathways which you can get this short chain fatty acid from protein. So we were interested in whether humans had more or less ability to produce butyrate compared to other primates. And what you can see here is that our ability is somewhat similar. So here in the purple, humans are always at the beginning, and then these other primate groups are off to the right, or it goes ape, Cercopithecine, Platyrrhine, which are the New World monkeys, and lemur. And you can see that humans don't necessarily have more or less compared to other non-human primates. What was interesting though is when we looked at humans that were eating more or less processed foods, kind of quote unquote industrialized versus non-industrialized humans, we associate industrialization and these processed foods in humans with lower short chain fatty acid production. And that is actually what we see right here with this fiber degradation pathway. But if you look at these protein degradation pathways, you actually see more in humans that are consuming more processed foods compared to the ones that are eating less. And this maybe makes sense as we shift our diet from fiber towards protein, that we actually shift the way we produce these short chain fatty acids from fiber to protein. And so in some ways we may actually be buffering our ability to produce these short chain fatty acids. We're just doing it a different way. And so this data set suggested that humans may actually have an equal or increased potential for producing this short chain fatty acid, and that we use different taxa and pathways to do this. But it's likely associated with an evolutionary shift in humans as a result of diet changes. So as we began to consume more protein as a species across our evolutionary history, we likely shifted to microbes that could still produce these short chain fatty acids, which may have been very important for our energy balance, but we yet just shifted to getting them from a different part of our diet. Now, there are potential health implications from this. Producing short chain fatty acids from proteins creates other byproducts that we know are associated with things like intestinal inflammation, colon cancer, etc. So there is a health risk from producing too many short chain fatty acids from protein, and it's still important for us to have fiber in our diet. So I think this shows us that the human gut microbiota has some unique properties that may be related to our unique evolutionary history and adaptations that are associated with our expensive life history traits and the fact that we live in variable environments. But we're really kind of just scratching the surface to try to kind of get here. I think the related question is, how did we get this gut microbiota? And I just want to make an important point here that I pretty strongly believe it's not co-diversification. So a lot of people think that this phylosymbiosis pattern between host and microbe comes from the microbes diverging as their hosts diverge. And the evidence that we've had from non-human primates is that actually in many cases, the divergence of these microbes predates the divergence of the hosts. So the hosts are somehow picking up microbial taxa that are more ancient than themselves, kind of preferentially from the environment. So we still see this species-specific pattern, but it's not because the microbes are co-diversifying with their hosts, it's because the hosts are acquiring these specific microbial communities in some other way. There are a subset of microbes that look like they're co-diversifying, but this seems to be the minority. If we look across the animal kingdom, it seems that there may be a relationship between microbial diversity and the immune system, and essentially kind of how strongly the immune system is controlling which types of microbes get in. So this is a figure from a review that I wrote a couple years ago now, just looking at whether or not we see phylosymbiosis in these different groups of animals. These are the green dots, so the darker means we tend to see it more often. And then we're associating it with kind of how diverse the microbiome is and kind of how the immune system is built. And it seems like this may actually be related to placentation in the mammals. The mammals, we tend to see more species-specific microbiomes. They have very rich microbiomes. And they have immune traits that are associated with having a placenta that require the immune system to be a little bit more specific in how it's interacting with kind of foreign cells, which include microbes, but it would also include the fetus during pregnancy. We do see phylosymbiosis in some of these animal species that tend to have less diverse microbial communities. These seem to be a result of passing those communities on directly, either intracellularly or kind of at the time of when eggs are laid, or when eggs are released into the water, etc. So this is a very different system of transmission of microbes that seems to lead to species specificity of the microbial community. And we think that as you get to these more complex microbial communities, then it relies more on the immune system. The immune system, and in mammals specifically, I'll also say there are other sorts of traits that we think facilitate this transmission. So things like microbes being transmitted in milk, right? Microbial transmission during birth and after birth. I already talked about the immune system here in the middle, and then also social interactions in these social groups where you can actually trade microbes between individuals. So it's a different system of microbial transmission, but this is how these microbial communities are probably being selected out from the environment rather than the microbial communities co-diversifying with their hosts. All right, so I think I will leave it there for now. I can talk a little bit more about some other themes if there are questions about it. But really what I hope to kind of demonstrate is that understanding this host ecological context and the evolutionary context really gives us better insight into host microbiome dynamics than it would if we were kind of just looking at it from a kind of health and physiology perspective. Now, like I said, I am really just scratching the surface here. I think there's a lot left to explore. We need to generate data from more wild animals in different contexts, right? I talked a lot about primates because that's what I have the most data from. But I do think that these dynamics are likely to be occurring at least in other mammals and probably more other types of animals. I think we need to dig deeper into what makes the microbiome of different animal species unique and try to understand how those unique microbiome traits associate with the unique host traits in ecology and evolution. And kind of try to understand which traits may actually be in the microbiome are contributing to those host traits via kind of being a symptom of them, right? Kind of which direction is the causality. As well as taking into account other factors, right? A lot of times people with kind of these newer tools and technologies want to use it to kind of describe everything. And while I think the microbiome is very important, it's clearly not the only thing that's important in dictating host health, ecology, or evolution. And so really trying to understand better when is the microbiome actually a more important piece of this puzzle and when is it less important, and kind of, you know, what host species does that vary across, what environments does it vary across, etc. So with that, I'd like to thank my lab. I have 2 pictures on here because there were people across years involved in these studies, and my picture is always old and missing people. First and foremost, also, I would like to thank many collaborators, co-authors, field assistants, and permitting agencies around the world. I can't list them all here, but they are listed on all the papers that I had at the bottom of my slides, as well as many funding agencies for supporting this work. And I'm happy to take any questions.
Thank you, Katie. Excellent. Let me see if I can come back. Let me see if I can. Oh, I don't know. I don't know why I'm not getting there, but standard. Okay, I'm back. So if anybody, we have already 2 questions on chat, but if anybody wants to ask in person something to Katie before, we can now open for questions. I have a couple also later.
I have a couple, but we can wait for the ones on in the chat. Can you enable my video again?
Okay, yes, please.
It's up to you actually to enable the video.
Are you sure? Let me
Yeah,
see.
it says I can't come back. The
Oh,
host has
okay.
disabled the video.
I like this power. Ask to
Yeah,
start
it's
a video.
powerful.
Okay,
Right.
now I asked you to start. Okay,
Thank you.
the way that I like.
I'll wait for the other questions, but yeah, it was really good.
Okay, I'll then ask the first one from Landa Miles. Thank you for the presentation. It was very interesting and enriching. Congratulations. And she asks, is it possible to identify species by their microbiota fingerprint? If so, should we consider the season of the sample?
That's a great question. Yes, we can. I actually have an instance of that where I had suspected we could, and then I got to test it. I actually, on that 18 species comparison, I got samples from many different people, and some people contributed samples from multiple species. And when I put it all in a plot, there were, these were colobus monkeys and red tails, and there was, I believe it was a red tail that was clustering with the colobus monkeys. Like there was a point that I had colored based on what I had been told, but it was very much clustering with a different species. And I said, I think somebody mislabeled a tube. And we had shotgun metagenomic data, which gets you DNA from everything that's in fecal. So there is a little bit of host DNA in there. And so we actually went in and pulled the host DNA reads out of that sample and checked them against a database. And we found out that indeed the sample had been mislabeled and that the microbiome data was correct and that it was a different species. So yes, you very much can identify species based on their microbiome as long as you have some, you know, database idea of what the microbiome should be. In terms of season, that does not seem to matter too much. The effect size of host species tends to be much greater than the effect size of season there. So I have samples from howler monkeys in the same forest as spider monkeys, and it's a different howler monkey species with the same spider monkey species in 2 different countries in the forest. And the howler monkeys always cluster together, even though some of them were in the same forest as some of these spider monkeys. And it doesn't matter if the howler monkeys were eating more or less fruit or what they were eating, they always clustered together as howler monkeys, and the spider monkeys always clustered separately as spider monkeys. It probably gets harder as the species become more related, right? It might matter more if you were comparing two different howler monkey species in the season, you might start to get more overlap. But generally speaking, it's a pretty robust way to identify species, and you don't have to worry so much about the other environmental traits.
Juan Carlos wrote, "Dearest Katie, thank you so much for your always outstanding conference. Do you have any modeling predictions or studies that relate this kind of microbiome findings to the future effects of global climate changes and how the microbiome will have a major or not influence on the distribution of new primate species worldwide? Felicidades, querida Katie."
I don't have great data. I think the closest data that I have are the ones from the two howler monkey species that have different distributions. But there is some speculation involved with that. My hypothesis is that it will have an impact in terms of seasonality of food availability and how well the microbiome can shift in response to this, whether the microbiome can shift sufficiently or not, right? Or if these diet changes are going to be too big and too fast and too long term for the microbiome to be able to kind of buffer against them. I also suspect that we are going to start to see the microbiome becoming very important in the related context of plant nutritional content or nutritional composition, right? In terms of thinking about our plants starting to have more fiber, more plant secondary metabolites, right? As their environments become more difficult, are they going to start to defend themselves against herbivores more because it's so costly to produce tissue for themselves? If we're starting to see more of those compounds in the plants, those are things that the microbes are really helping their hosts deal with. And so again, does a host species actually have the microbial community it needs to be able to deal with that change in the food composition? Or is it again going to happen kind of too quickly and in ways that can't be kind of buffered quickly enough by the microbiome? The microbiome is more plastic than host genetics, but as we saw, it's still relatively constrained. I think a lot of the human research is very focused on diet can change the microbiome and we can change it so easily. But if you really zoom out and take this cross-species approach, there is flexibility, but there's also very clear constraints. And so it's not that easy to kind of just change the microbiome. So I think that's a good question. And I think, again, getting more information about these diet microbe interactions in wild species and trying to understand what actually is happening and in which context is going to be important. And I think more studies on species where you have differences in geographic distributions, but very similar ecologies, and trying to understand if microbes might be playing a role in some of that, I think are going to be really important. Those can be hard studies to do because it requires sampling across, time and space in a big way. But I think we need more data about that to be able to kind of back up my hypotheses.
Duda, you can ask your question or your questions.
I have several, but I'll just pick a couple.
So we can.
Yeah, it's great. So congratulations. It's a very nice talk and very nice set of studies. I'm a professor of evolutionary biology here, and I work mostly on carnivores and carnivore and evolutionary and conservation genomics. And so I don't follow the primate literature that closely, but I probably should because it's pretty cool. And although we have Julio here, I still see some papers come out, but not enough. So a couple of questions. When you mentioned cercopithecines, there were just a few species included, right? And that convergence between humans and cercopithecines is really interesting and intriguing. Do you think that is influenced by what cercopithecines you included? Because there are some folivores, right? You have colobine and langurs. So you did have mostly omnivorous cercopithecines probably in that data set, right?
That's correct. So you see, can I get back there? Well, actually, I don't have the big, yeah, I don't have that one graph. The colobines, yes, so the leaf-eating cercopithecines, they don't cluster as much. You are correct. They have a very different gut physiology, right? They have sacculated foreguts like a cow. And so their gut physiology is very much different. And that was that 18 species comparison, you know, that's been shown across mammal species. There were like some big science papers like 10 years ago about, you know, diet drives the microbiome. But if you look at them, they're actually confounded by phylogeny to some extent and physiology. So a lot of their herbivores are ungulates. And they're things that tend to have a similar digestive physiology. There's one about ant-eating animals as well. And that's phylogenetically less confounded, but when I actually looked it up, I didn't know this, but it makes sense. Apparently all ant-eating animals have a specialized stomach that can grind chitin. And so they are very similar physiologically. When we look at the primates, leaf eating has evolved several times independently with a very different physiology. So those leaf eating primates with the sacculated foregut have a very different microbiome than like a howler monkey that just has a slightly enlarged hindgut. And so I think a lot of these diet patterns that people are picking up on are actually physiology patterns. And people are kind of confounding diet with phylogeny and physiology. Like diet is kind of a dietary niche, I think is in some ways a proxy for a lot of other physiological things that are probably more directly affecting microbes. So back to your actual question, yes, you're right. It does depend on which species we're looking at. You know, those primates with the sacculated foregut, they are doing something very different. Within the cercopithecines that are actual cercopithecines that have cheek pouches and they don't have this sacculated foregut, they are generally all showing the same pattern, although I will say that the baboon specifically is the most similar to humans in that data set. We started to play around with macaque data that were publicly published, or yeah, that were public, and we didn't get to the point where we could publish on it, but rhesus macaques have an even more similar microbiome to humans. So yes, I do think it depends to some extent which primate species you're looking at.
Well, yeah, because it looks like it's a great system to assess convergence, right? You can have multiple families there and multiple life histories, and you're going to have probably more instances of convergence than just one, right? So it's you can have the leaf eating, well, not considering that complexity of the anatomy and physiology, but if you could control for that, that'd be really neat. And then just another question is regarding individual variation. What I understood from your early slides is that you have a lot of samples per individual, and you can tell the individual, right? And
Yes.
that's a kind of a very interesting. So all those early seasonal patterns were individual based.
Yes.
Yeah, so it's quite interesting. You have different scales, right? You can look at the individual along the time. You can look at population level things and across populations, across space, across time. Yeah, I can tell while you're busy with the primates. It's cool
Yes,
stuff.
they keep me busy. Yeah, and I haven't looked so much closely at the individuals, but I have data that are of course getting old now, but they're still cool, where we are trying to put together microbial genomes. We have very deep sequencing from those individuals, and we sample them every 2 weeks to kind of look at that change every 2 weeks. You know, can we actually track? We have the diet data. Can we actually look at those patterns? So I just have to get somebody that can help me move it faster.
Oh, one last question, sorry. What about eukaryotes? Are you typing 18S as well and looking for eukaryotic communities?
We did that. So with that big 18 species study, I handed the DNA over to somebody that was doing 18S because they were better at the analytical pipeline. And we looked at it. We don't see the same strong patterns in terms of phylosymbiosis or convergence. So we published the data, but the story was a little less clear. I know that there are like Tony Goldberg at University of Wisconsin-Madison has developed a new pipeline that he thinks may work a little bit better, at least for the parasite side of eukaryotes, at picking some more things up. So we've dabbled a little bit, but I don't have anything super exciting that has come from that yet.
Well,
Yeah.
let's see what the future holds, but really cool. Congrats.
Thank you. No, thank you very much. I guess the other thing I'll briefly add is that we've actually replicated the human cercopithecine convergence with a completely different like set of samples. And so it happened again, you know, you're always like, and we got it again. And this data set, people have often asked me about. Some chimpanzees live in a savanna environment currently, and some baboons live in a more forest. And so like, would that matter? And preliminarily, it seems to not matter. So I think this is more of an evolutionary thing. A savanna chimpanzee still looks like the forest chimpanzees and is more different from humans.
Interesting. Yeah.
Okay, thank you, Katie. I have a few questions.
Sure.
I'll begin with one that you showed that the humans have a much larger, wider inter-individual variation. And I was curious to know if those where those individuals are, if they are also spatially spread, or if they are from the same populations and or they have the same diet. So what are driving those differences in humans, considering that we are widespread throughout the world? It's diet, it's environment, it's location. What is the major driver that you believe?
That's a great question. So they come from a more, generally speaking, more spatially diverse array of places. So some of them are from, I'm trying to think, there's some from Tanzania in there. There are some from South America. So they're different continents, mainly Africa and South America. What I think is driving that is probably kind of local microbial adaptation to diet and lifestyle. I think, you know, it's interesting if you look, you know, there's still, when you compare them to other primate species, right, they're still all clustering together here. But yes, when you kind of look at how different they are from each other, it is different. And so I do think those are kind of local adaptations. Diet is the thing that is often talked about in the human literature. But it's a little bit oversimplified because there are things, a lot of these are cross country, cross population, and there are many things that differ besides diet. And none of these variables historically in the microbiome literature so far have been well documented. So people tend to go in and get fecal samples, but they're not getting the dietary data. They're not getting data about like outdoor exposure, social interactions, other things that we think might affect the microbiome. I think studies now are starting to do a better job of that. But in those data, we didn't have them so much. So yes, I think it is kind of local adaptation to environment, but we need better data.
Okay, thank you. I was also thinking about the data from Janaína's thesis. She's somewhere here. She's hiding here. And consider that the seasonality of the environment may drive the gut microbiota, at least within a species and within a population. Should we, and I don't remember what we found actually, so I'll ask you now because I thought if we find something that I thought that we should expect, should we expect more stable microbiota composition and the relative abundances of the taxa in the supplemented groups compared to the non-supplemented groups?
Yes, that's a great question. So, so we did find seasonality in the data set, in both groups. So these are howler monkeys that we're getting for everybody else. The howler monkeys in the wild, some of them are being supplemented by humans with human foods. Some of them are not. We did see an effective season regardless. I think the effective season is slightly stronger in the non-supplemented groups, but it was still a significant effect in the supplemented groups. So the supplementation, at least in that population, doesn't seem like it's enough to completely counteract the seasonality of the rest of the diet. Because my understanding, based on Janina's data from the feeding data, is that even those supplemented howler monkeys are still eating a fair amount of their wild diet. So I think what's happening there is that, yeah, there might be a little bit less seasonality, but there's still enough seasonality because they're eating enough wild foods that we still tend to see that dynamic. So yes.
Excellent. And also related to this in terms of diet stability or just eating the same diet over time, I was curious if you have data on captive individuals and if is it possible to see extinction of some microbiota taxa because they are not being useful or not right, or they are not, there is no input through the diet or something like that by eating in captivity a more stable diet over time throughout every day and so on.
Absolutely, that's a great question. So I would say primates, but also mammals more generally, we do see an effect of captivity on the microbiome. It tends to be stronger in dietary specialists, so things like leaf eaters. You do see a big reduction in diversity in captivity. It looks like it's not affecting omnivores quite as much, probably because, again, their microbiomes kind of do everything, and their diet may not be quite, pardon me, as different in captivity as the specialists, right? Leaf-eating primates in the US don't get a lot of leaves in captivity because we just don't have them, right? So they get like fiber-based monkey chow, which I'm not sure how much fiber is actually in that chow. There was a really cool mouse study several years ago where they had mice in a lab, and they actually took fiber out of their diet and then bred them across generations. And what they found was that as they did that, they actually did lose the microbial taxa that were associated with digestion of fiber. Perhaps not surprisingly, they introduced fiber in like generation 3, and that did nothing because the microbes were gone. And so they showed that you had to actually reintroduce the microbes and the fiber to be able to kind of rescue that microbiome. So that does occur. Granted, that was a very controlled environment where other microbes were not coming in. So I think that sort of response would probably be slower in the wild, where you have exposure to other animals and you're not kind of in a sealed environment. But yes, that absolutely does seem like it happens.
Excellent,
And
thank you.
yeah, and that's actually a concern many of the human microbiome researchers have about the human microbiome, is that that's what we're doing to our microbiome as we eat more processed foods with less fiber.
Yeah, there are some infections that we can lose a good proportion of our microbiome, and we need to have some new input from somewhere, right? That I remember a colleague of ours that was a parasitologist telling some interesting stories about using the partner sometimes to as a donor of microbiome or something like that. So it's
Right.
interesting.
Yes, yes. It's interesting though, and I have to look more closely at this data now that I think there's more out there, is that you don't see the same patterns in indigenous people in the Arctic that tend to have a low fiber diet traditionally. They tend to have a microbiome that also still looks like the non-processed food, kind of more of some of the populations that eat more fiber and hunter-gathering kind of thing. And so I don't know that we have completely figured that out yet. I was thinking about the carnivory. So.
Good. Do you want to make another question, Duda, or somebody else that is listening and shy to
No, I think I'm
either?
good. I'm thinking of the carnivory too. I mean, I work on
Yeah,
carnivores. Some of them are omnivores. And yeah, lots of cool questions there.
excellent. Okay, if nobody else has any other question, there is nothing new in our chat. And I think that's excellent. It was a great talk, a very nice set of questions, wonderful answers. So, Katie, thank you very much.
Thank you for having me.
And it was really great to have you here. Then I'll later upload in our YouTube channel so more people can benefit from your knowledge and everything that we have seen today.
Excellent.
Thank
Thank
you.
you so much. Thank
Thank you
you.
very
Thank
much. It was
you.
great.
Bye bye, everyone.
Thanks. Bye
Bye
bye.
bye.
Bye.